Dear participants, dear guest speakers, let's start our next session, which is focused on South-to-South collaborations on food security and innovations focused on ICGEB. I'm really happy to be able to participate in this very important convention, and I would like to thank Dr. Musomi and his team for inviting ICGEB to run this session. We have 4 guest speakers today. All of them are group leaders from the ICGEB. The first one would be Dr. Dennis Indolo, who is the group leader of biopesticides at the ICGEB Cape Town component. One of the major activities of his group is the biopesticide residue mitigations and regulatory harmonization project in Southern Africa. Dr. Indolo, the floor is yours.
Thank you very much. Okay, I'm going to talk on the potential of using biopesticides to enhance markets for African agricultural produce. I'd like to, sorry, I may need some technical help here because, okay, sorry, it's moving. Yeah, how to promote the strategic use of biopesticides to enhance market access for African agricultural produce. Now, there has been a decline of African food exports over the years. Before 2000, Sub-Saharan Africa actually was a net exporter of food. But now, Sub-Saharan Africa, which is home to 13% of the world's population, has a net food trade deficit of $7 billion US. And it accounts for less than 3% of global agricultural exports. In fact, Sub-Saharan Africa exports less food than Thailand, which is a country that accounts for only 1% of the world's population. Now, why is this? There may be a number of reasons why Africa is struggling to sell its food to markets out there. And one of these reasons is because the produce from certain countries has got higher than acceptable chemical residue limits. So for example, in the European market, in the EU, 7.6% of agricultural produce exported into that market have found to have higher than acceptable pesticide residues, what we call maximum residue limits. As opposed to imports from within the EU, those exceed MRLs by only 2.6%. And what happens is that if imports are found to repeatedly exceed these limits, then they are placed on a list of imports that require further controls, and those countries can stop importing from countries which regularly send them produce whose pesticide residue limits exceed acceptable amounts. So that is one of the problems why some countries are struggling to access these markets, because they use synthetic chemical pesticides, but by the time they're harvesting, the produce still has a lot of chemicals, and when it's tested out there, it exceeds the amounts that they would accept. So what I'm showing on that slide is a report that actually was done by the European Union, and that's where the figures are from. Now, there's also data showing that pesticide usage in Sub-Saharan Africa is still quite high and is actually bound to increase. For example, if we have high growth in the agricultural sector, pesticide usage is expected to increase from about 0.12 kilograms of active ingredient per hectare to 0.27, that's what's shown in red there, over the next 20 years. So despite the fact that some countries are struggling to get into markets because of high synthetic chemical pesticide residue levels, the data still shows that pesticide usage is still bound to increase, and therefore problems of accessing markets are bound to stay with us for a while unless that issue is addressed. Some other statistics shows, for example, if we talk of Southern Africa, if we look at it at a regional level on the continent, Southern Africa has the highest pesticide usage on the continent. That's what's shown by the red arrow. So you see that graph, the red arrow shows pesticide usage in Southern Africa. The curves below it show pesticide usage in other regions in Africa. So Southern Africa has very high pesticide usage, and it's also expected to increase. Over the next 20 years, pesticide usage in Southern Africa is expected to increase to almost double in the next 20 years. As I said earlier, this is also a publication that talks of pesticide use in South Africa. South Africa is actually one of the largest importers of pesticides in Africa. And as I said earlier, the financial impacts of sending to external markets produce that does not meet the requirements of the importing countries is very significant. For example, South Africa exports a lot of citrus. And if, for example, citrus from South Africa exceeds the pesticide residue levels in the markets to which they export, the country has a potential to lose up to $54 million US. Now, previously, there are certain chemicals, synthetic chemicals, which have been detected in produce from the country, in grapes, in pears, in pepper, chemicals such as dimethoate and ethaphone. But the reason I put this slide up there is just to show that the consequences of exceeding MRLs is very significant. And if a country's exports are stopped because of exceeding MRLs, it takes up to 4 years to try and sort out the issue to get that country exporting again to that market. So how do biopesticides come in? Biopesticides have the potential to mitigate this problem. We have a number of scenarios. One, farmers can either use only pesticides to control pests on their crops. If farmers use only synthetic chemical pesticides, they get high yields. They get very good yields. Their crop quality is quite good. I mean, the visual quality, so there's low crop damage. But the only challenge with many crops is that if you use synthetic chemical pesticides throughout the season, then by the time you're harvesting the crop, the chemical residue levels are quite high, and then you have a problem accessing markets. So that's scenario one, where a farmer uses only pesticides throughout the season to control pests. The other alternative would be a farmer may decide, okay, we have problems with chemical pesticide residue levels. So a farmer may decide that, okay, for me, I am going to use biopesticides throughout the season. Now, if that happens, what happens is that biopesticides do not provide as much control. Their efficacy is not as great as for synthetic chemical pesticides. So a farmer who decides to use only biopesticides will get low yields because they are not as effective as synthetic chemical pesticides. The farmer will get high crop damage and hence poor crop quality. The chemical pesticide residue in the crop will be low because biopesticides are generally made from, say from microbes, for instance. So they do not leave chemical residues on the produce. And also, exporting countries normally don't have any requirements for testing of biopesticide residues. So they'll have low residue levels, but they will still have difficulty accessing markets if they use biopesticides alone. Why would they have difficulty accessing markets? It's because consumers are interested not only in the fact that the produce is free of chemical residues, but also the quality. Very few people would be willing to buy a crop that shows a lot of pest damage. So much as those crops will have low residue levels, these farmers will also have difficulty accessing markets. The last scenario is a case where farmers use pesticides for early season pests. So early in the season, the farmers use the conventional synthetic chemical pesticides. But then late in the season, for those pests that come late in the season, the farmers then use biopesticides, because what happens is that when you stop using pesticides at some point, it starts degrading. So in that scenario, the pesticides are able to provide effective control early in the season. The biopesticides are able to provide some level of control late in the season. The farmers are assured of high yields. The farmers are assured of low crop damage. And because they have used biopesticides towards the end of the season, the residue levels are low and the farmers are hence able to access markets. That is the point of my talk, that used strategically alongside conventional synthetic pesticides, biopesticides have great potential of enhancing market access for produce from African countries. And on that note, the International Center for Genetic Engineering and Biotechnology is implementing a project that is working on trying to enhance trade through promoting the use of biopesticides for late season pests based on the principles that I've outlined in my talk. I will leave it there. That is the link to the project. It's being implemented in Southern Africa, and it's essentially trying to exactly promote the use of promote the strategic use of biopesticides exactly as per the principles outlined in that talk. Thank you very much for listening.
Thank you, Dr. Indolo, for your nice presentation. Our second guest speaker is Dr. Vittorio Venturi. He is the group leader of plant bacteriology at ICGEB Trieste and also the scientific coordinator of ICGEB. His research is focused on the interkingdom signaling between plants and plant-associated bacteria, and he uses as applied research. This is specifically aimed to find microbial solutions for sustainable agriculture. Dr. Venturi, the floor is yours.
Thank you, Luis. I will now share my screen. So, hello everybody. It's a pleasure to be part of this session. As part of ICGEB, we are an organization with 3 institutes, with 3 components: one in Trieste, one in New Delhi, one in Cape Town, South Africa, and over 60 member countries. And as Luis mentioned, I am located in Trieste, in Italy, and I am part of the mandate of ICGEB is to do training of young scientists coming from our member countries and also promote biotechnology. And I'm particularly focused on how bacteria live and colonize plant environments, and also how they can be used to improve plant health. So, you know, newest technologies, especially in next generation DNA sequencing, has allowed us to document that basically every living organism, you know, humans, plants, live together with so much microbiology. So many studies have been done on humans, and in fact, humans have 10 times more microbial cells in their bodies than human cells. Research in the microbiology of plants is now catching up. And what we're also seeing in plants is that plants live together with a lot of microbes. And microbes are located basically everywhere, on the fruits, on the leaves, and also inside. But most importantly, what is really the most important region of the plant is the rhizosphere. And that is the soil, you know, up to 2-3 millimeters away from plant roots. Okay, so this can really be paralleled with the human gut. As we know, we humans have a very rich microbial life in our gut, which provides, you know, very important functions. In fact, our microbiology of the human gut is like an external organ, and the parallelism here is similar. So the microbes that live very close to the roots is very rich and provides a lot of important activities for plant health. Okay, this life, microbial life, the most important driver of this microbial life is the soil. So it is very important that the soil is rich in microbial life. And this is a very important topic also, the Food and Agriculture Organization that is launching, that we are using, our soils are very much exploited in intensive agriculture. So a major issue in the future is to keep our soils healthy in terms of the microbiology. Okay, so we are very interested to study the microbial life of the rhizosphere, so of the soil closest to the roots that the plant recruits actually. So this microbial life near the roots in the rhizosphere provides so many vital functions for the plant, and they're shown here. You know, they improve the nutrient uptake. They help the plant through abiotic stresses like heat, high temperatures, or low amounts of water, high amounts of salt, and also biotic, provide disease resistance. And also they can promote growth of the roots because they produce plant growth hormones, and they also induce immunity. So studying this microbial life can provide very important alternatives to what also Dennis Nodolo was just saying, to the use of chemical fertilizers. If we can use as much as possible the microbiology of plants, we will be in a position to reduce the use of fertilizers as well as pesticides, because we can use the natural microbiology to make the plant stronger. So we've been launching together with scientists in other ICGEB components in New Delhi and in Cape Town. We are now just launching a project on sorghum. Sorghum is by far the most important African cereal. The reason why it originates in Africa, it's a very resilient crop. It's drought and heat tolerant. It's, you know, particularly sorghum needs a lot of nitrogen, and it's particularly nitrogen efficient. And it has various uses. You know, it can be used as human food. Most importantly, used a lot as animal feed, and also is used in several beers and also biofuels. And this is a topic also that my colleague, my next speaker, Shams Yazdami, will introduce to you. So as you can see, it's a very important crop for Africa, and it's grown in many African countries, most notably in Sudan and in Ethiopia and Nigeria, but many others as well. So what we intend to do in the next few years is to understand as much as possible the microbiology of sorghum, and to try and understand and isolate the most important bacteria, beneficial bacteria which live together with sorghum. So what we will do is we will study the complete microbiology of several sorghum varieties that are grown in different farming practices, and also sorghum plants which are diseased and healthy. And all these photographs of microbiology, we will compare and study and analyze them carefully in order to understand which are the most important bacteria that associate with sorghum, so which form beneficial associations, both in terms of sorghum recruiting them and bacteria and bacteria being able to communicate and live together with sorghum, and then isolate them in order to devise and you know, the long-term goals is to make products, microbial products that can be used to control diseases or to reduce the use of fertilizers, because applying these probiotics will give an advantage to the sorghum, thereby hopefully reducing the use of these chemical inputs. Okay, now there's a very important world interest in microbial solutions for a more sustainable agriculture, because basically we cannot afford to continue to pursue intensive agriculture as we do today. The microbial inoculants market in agriculture is now valued at approximately $4 billion, and it's increasing by 10% every year, because governments are under a lot of pressure to reduce the use of chemical inputs, both in terms of for environmental issues, but also because it's not sustainable. We cannot keep on producing fertilizers and pesticides, synthetic fertilizers and pesticides, because basically the raw materials are decreasing, and the earth cannot provide them at a continuous basis. So it's a very important field, and many startups are being created. Many companies are dedicating money in trying to use this microbiology information in order to devise ways to decrease the use of chemical inputs. So what we're doing now, and what we started to do together with Dr. Lauren Donaldson, who will be the last speaker also of today, we are creating a network of scientists in Africa. As you can see here, we've contacted our scientists who are also very interested in understanding the sorghum microbiology. And you can see with different institutions in Africa. So we will team up together, and we will sample different sorghum varieties under different farming conditions, under different health status, different abiotic stresses, and all this information will be analyzed. And then also we will be interested in isolating microbes. And I think what's an important point here is to isolate microbes locally. So, isolate the different countries in Africa can make their own culture collections and devise their own beneficial microbes and collaborate with local industries to develop microbial inoculants for sorghum. So we made this network, and now we are starting to sample and to determine the microbiomes of all of different sorghums in Africa. And I also this. It's a pleasure to do, but you also have to appreciate we do things respecting the rules of the Nagoya Protocol of Access Benefit Sharing. So we have to make sure that all the information will be accessed and benefited by all the countries that will contribute to this knowledge. Finally, this is my last slide. I'm involved in a major project in coffee, which we're doing exactly the same thing, looking at the microbiology of the rhizosphere of coffee plants in different parts of the world. And so in parallel, we're also, the science is very similar, the concepts are very similar, so we can very quickly change crop or plant in order to do similar experiments. So this is the end of my presentation, my brief presentation, which and this is my email, and I'll be very happy to talk to any of you who are interested to have more detail and more in-depth discussions, of course, in this topic. Thank you very much.
Dr. Venturi, for the nice presentation. We will have some questions in our Q&A section. Our next guest speaker is Dr. Shams Widzani, who is the group leader of Microbial Engineer Group at the International Center of Genetic Engineering and Technology in New Delhi, in India. His interest is shifting to non-food-based feedstock for biofuel production. Shams, the floor is yours.
Thank you very much, Luis, and the BioAfrica Convention for inviting me to present on this very important platform. It's a great connectivity for all of us. And I'm basically a group leader of Microbial Engineering Group at ICGEB New Delhi. However, in the microbial engineering, my main focus area of research is biofuels. And I'll just take you through that little bit of the journey that we are into it. So the reason why we went into this area was mainly because of the impact that fossil fuels are creating in our environment. And you can see that there are 35 billion metric tons of carbon dioxide that are being annually emitted to the environment, and 3/4 of which is contributed by the usage of fossil fuels. And these are the countries which are a major contributor of the CO2 emission. We can see some of the countries listed here, the China, India, Africa, South America, all of them are being increasingly involved in emitting this. So we need to find an alternative for this. And the outcome of this carbon dioxide emission is basically a global warming, which is leading to a very, very severe weather condition, rising of sea level, rising of temperature. And overall, it is impacting not only on the pollution and other aspects, but the human health assets. So everything is basically linking together, and we are all getting impacted largely because of these pollutant that we are secreting in the environment. So it's very, very important to find an alternative means so that we can stop the release of the global warming gases. So obviously, biofuel is a very good alternative that we have for the energy. And we hope that we can reduce the emission just because the biofuel is a carbon neutral energy source. Because you can, within the year, you can recycle the carbon within the environment. And therefore, it's very important to consider this instead of the fossil fuel itself. Now, when we consider the biofuels, there are different types of biofuels, so first, second, third generation biofuels. So first generation biofuels are mainly made from the edible portion of the plant, like cornstarch or the sugars coming from the corn or the vegetable oil. So of course, these feedstocks are competing with the food. So in terms of the food security, this is not really a good feedstock to start with for making fuels like bioethanol or biodiesel. And therefore, we focus at ICGEB New Delhi on second generation and third generation biofuels. Second generation basically focuses on the non-edible part of the plant biomass, or from other plants which are not competing with the food, while the third generation biofuel actually look for algal as a feedstock for making biofuel. Now, in India, we found that the nearest biofuel that we could achieve in terms of the second generation are from the agricultural residues, mainly because agricultural residues are present in surplus. India being a major agricultural country, we produce almost 500 million tons of biomass every year. And we believe that majority of them are not utilized properly. A lot of government data suggests that, and those which are not utilized properly can be converted into fuel like ethanol. Obviously, there's government effort to make this happen, because I will show you that this process of making fuel from agricultural biomass are expensive, tedious. So there's a lot of biofuel policy which has been made to provide a tax incentive or the viability gap funding, so that more and more industries, companies, venture capitalists, they come forward to invest and produce biofuels. They also increase the purchase price of these. Another reason, particularly in India and other places like India, is that a lot of these biomass are burnt in the field itself to clear the field quickly, and they add pollution in the air. Therefore, there is a need to identify routes which can produce biofuel from lignocellulosic biomass. So this is a simpler flow diagram to suggest how to make a biofuel from the lignocellulosic biomass. And you can see that agricultural residues are made up of lignin, hemicellulose, and cellulose. So we need to do a pre-treatment to get rid of the lignin, so that these structural polymer of sugar, hemicellulose and cellulose, are accessible to the enzyme. And then after the enzymatic hydrolysis, they can be fermented to fuel. So there are different challenges. Every step is challenging because the enzymes are very, very expensive because of their low efficiency and the low titer. And that became one of the major focus of our study to improve the enzyme efficiency in the titer. The agricultural residues also have a lot of C5 sugar, which is not fermented by yeast. So that's another thing that we work on. And then the ethanol being only commercial fuel at this moment, but it's not very energy dense. So we also work on other energy dense molecules like butanol, long chain alcohol, and alkane. So in terms of enzymes, we started focusing on fungal enzymes because fungi are known to produce a huge amount of secretory proteins. So we started screening the microbiome of all the Indian flora, as well as some of them coming from the culture collection. So we were funded by the Government of India robotic system and all the screening facilities so that we can screen a large number of isolates. And we identified one of the fungi which was producing a very good efficient enzymes. We further characterize through the genome sequencing, proteomics, and structural function relationship about the details of those enzymes so that we understand how to improve them further. We identified the reason for some of these enzymes being more efficient than the counterpart which are readily available in the market, and a lot of kinetic modeling also suggested the superiority of this. We use one of these fungal hosts as a platform to do genetic engineering, the Penicillium Funiculosum, because we found that it has the ability to produce good enzymes. So we developed tools to manipulate the genome through all the advanced systems that are available to do the synthetic biology kind of approaches. And we were able to delete the repressor and overexpress enhancers and many other enzymes to make it much more efficient in terms of the efficiency and titer, which was actually looking at the efficient production of these enzymes and its enhancement as compared to the commercial enzyme, best enzymes available in the market. The government of India decided to support the scale up of this particular enzyme technology, and we were able to scale this up to 15,000 liter scale. So we have a complete process starting from the screening to scale up to the commercial level at this moment in place. And of course, the patent was important. So we filed the patent at various countries. A lot of patent has already been granted, like US patent has been granted on this. So now we are talking to industry of second generation biofuels to use these enzymes. The other thing I discussed was the C5 fermentation. That's another thing that we look at it very, very seriously, so that both C5 and C6 sugars are utilized effectively. We have developed the technology to do that. And here we can see that the C5 sugar has been utilized and ethanol being produced. And being the novel technology, the US patent has been granted on this. The other advanced fuel that we look at are butanol. Butanol is a better energy dense molecule. We do various kind of engineering to make the bacteria produce higher level of butanol through the CRISPR-Cas9 technologies and other type of engineering that we do to make it much more efficient production. The other important fuel molecule that we work on is a hydrocarbon itself. So we believe that all the fossil fuels are eventually hydrocarbon, carbon and hydrogen. So we focused on that, like pentadecane and heptadecane, which can be used as a drop-in fuel. So we can just put it in the engine, and we can see how it can be, used to replace the petrol or diesel. In between, we also produce another fuel molecule. We call it as a cetyl alcohol, which is closer to the hydrocarbon, just that it has a functional group as a hydroxyl group. It's an interesting molecule because it is also used in a cosmetic product, or the facial cream will have the cetyl alcohol. So this is also something that has been made possible through the effort of systems and synthetic biology. A lot of modeling has gone. So we do a lot of modeling to improve the yield and productivity of the product that we are working on. And then we do a synthetic biology to, for example, this E. coli has a 8 gene deletion and 3 gene addition to make this fatty alcohol and alkane produced at the highest quantity that has been reported so far. So most of these technologies, because of their commercial implication, has been patented. Many of them have already been, the patent have already been granted. Some of them are in the pipeline. We are working with the industries. We've already done a couple of tech transfer to the industries, and we are in the process of doing more and more transfer. So the important thing is all this effort on the biofuel area, the way we work in is need to go beyond the labs so that it can be actually useful to the society. So at that time, I'm stopping here and would like to thank all of you to giving me this opportunity. Thank you very much.
Thank you, Shams. Thanks very much for the nice talk. Our next guest speaker is Dr. Lara Donaldson. She's the group leader of Plant System Biology at ICGEB Cape Town component. Her research focusing on understanding how plants sense, signal, and respond and adapt to their environment. Lara, the floor is yours, please.
Thanks. So, okay, thank you to the organizers of the BioAfrica Convention for giving me the opportunity to speak to you today about the work that we're doing at the ICGEB in Cape Town. And it's a pleasure to be talking to you today about something I'm very passionate about, food security. So I recently attended the UN Summit on food security, where they presented the results of the SOFI report, the State of Food Security in the World. And I don't think I need to go into this too much, but basically a third of the world's population already doesn't have access to adequate food. And it's particularly alarming to see the numbers of how many children are malnourished or experiencing the effects of stunting and the long-term ramifications this is going to have on their development and the health systems. And we're certainly not on track to meet the Sustainable Development Goal of zero hunger by 2030. Why is there so much food insecurity? One of the reasons being that our food system is basically unsustainable. So we know that agriculture is one of the worst contributors to greenhouse gases, and this is largely because of the production of nitrogen-based fertilizers and pesticides. And after these are put onto the soil, they also then end up polluting our river systems and polluting the environment. And also agriculture is a major consumer of our fresh water. So here in South Africa, which is obviously a water scarce country, agriculture takes 70% of our fresh water. So by 2050, there's going to be another 2 billion people on the planet, and we need to be able to produce food for them. And in order to do this, we're going to need to be able to produce more with less inputs, and we need to do this in the face of climate change. I guess the traditional approach from plant scientists is to try and identify the genes that are involved in plant growth and plant stress tolerance, with the idea that we're going to be able to manipulate these genes to make more, either through genetic engineering or through selective breeding or through newer approaches such as CRISPR, to be able to make our crops more tolerant of stresses or be able to yield more in the future. And, as a plant scientist, we normally pick our favorite stress. So for me, I've done work, I'll show you on the next slide, on salt tolerance, plant salt tolerance. There's also obviously research on drought, flooding, and there's various biotic stresses as well that we're concerned about. This is just locusts. This is showing you some parasitic weeds that are devastating to cereal crops in Africa. And then there's also bacterial and fungal pathogens. I really think South Africa is a success story for why genetically modified crops do work. And there was a recent report in the Global Food Security Journal showing just how big the economic and ecosystem impacts have been for genetically modified maize in South Africa. Really massive increases in yield that has translated into many more food rations for food insecure people in this country. I think there's some hesitancy toward genetically modified crops, and it's quite sad, the story of golden rice, where we have a solution where rice has been engineered to contain beta carotene that can solve blindness, and this was developed not for profit, but to help the poor people of the world, and it hasn't reached the market because of policy and protests around it. But we still do this type of research. So for me, I do look at trying to identify genes that are involved in plant salt tolerance. That's my interest. And this is sort of, don't worry if you can't see all the detail in the slide, you can also check it out on the website. But basically, we've taken a transcriptomics approach to identify genes that are specifically involved in salt tolerance and not involved in other stresses. And the reason for this is because there aren't any salt tolerant crops in the market at the moment. And we think that this is because we haven't adequately identified genes that the plant uses to tolerate stress. And that's because salt stress is quite a complicated stress. So it imposes an osmotic stress in the plant where the plant can't take up enough water, but it also imposes a salt-specific stress where the sodium itself is toxic to the plant and competes for binding sites and uptake mechanisms with the essential plant nutrient potassium. So we tried to pull apart how the plant specifically responds to salt stress by comparing its response to salt and osmotic stress and looking for genes that really are specific to salt, and found that these genes are involved in this auxin pathway. So auxin is a plant growth hormone, and we think that maybe that's why plants can actually grow, maintain some level of growth on salt when they can't grow as big under osmotic stress conditions. And we've identified a gene that's involved in this auxin biosynthesis pathway, this nitrulase 2 gene, and shown that it increases specifically in response to salt. We see increased auxin levels in response to salt, and we can manipulate the expression of this gene, and we can show that we can enhance plant growth and survival under saline conditions. And we're busy trying to understand the mechanism behind that. But is this going to translate into a salt-tolerant crop for Africa? I'm not sure. So since I've joined the ICGEB, really got quite excited about this soil microbiome project that I'm doing together with Vittorio. And as Vittorio mentioned, in this project, what we've done is form this collaborative network of scientists across Africa. And the reason why we're so excited about doing the soil microbiome research is that research suggests that, yes, we can improve crop productivity if we make more resilient crops. But we can also do a lot by just increasing soil fertility. And if we can do both of these things together, increase soil fertility and increase crop resilience, then we can really get to where we need to be in terms of drastically improving crop productivity. And as Vittorio said, the plant microbiome is very important in enhancing nutrient uptake and improving resilience to various stresses. And so it can be argued that we can improve both fertility and crop resilience simply by manipulating the microbiome. And the microbiome has an important role to play in climate mitigation because the plants basically sequester, they capture carbon from the atmosphere and sequester and make it available to the microbes in the soil, thereby sequestering the carbon in the soil and acting as a carbon sink. So if we can manipulate this, we can, to make it a stronger carbon sink, then, we can mitigate some of the effects of the bad effects of agriculture, where we're changing it from being a carbon source to being a carbon sink. And I'm particularly interested in sort of expanding this project further beyond just looking at the microbiome, but looking at the interactions between the environment, the crop, and the farmer. And I think that some of the collaborations that we've made are going to be key to this. So I'm excited about working with Lisa Rothman at the University of the Free State and Edson Nkuba at the ARC, because they're already working with sociologists and farmers, and we can try and gather as much information about from the farmer and how what they're doing is influencing the microbiome. And similarly, Joseph Malema is working at CABI in Kenya, and they also work with extension officers with farmers, and we can look at how farming systems in Africa are affecting the soil and how this is changing crop productivity. I think looking at the systems approach, we need to do the basic science to be able to see where the gaps are and where the space for innovation lies. I'm looking forward to see where it leads us, whether it's perhaps we might be able to develop diagnostics for soil status, digital solutions, as my other colleagues have said, biofertilizers, biopesticides, understanding plant varieties. Also, I think something that's quite interesting is that, if there is an appetite for engineering crops, maybe there is for engineering microbes. But I think whatever we do, we need to basically engage all stakeholders to make sure that we don't end up in a golden rice scenario. So I think that, as Lawrence said in his address this morning, it's important that we work with outreach, with education, and work with the lay public to make sure that whatever the innovations that we come up with are accepted and adopted and make a difference. Yeah. I just wanted to keep short so we have some time for questions. Thank you. And I'm excited about our recent collaborations for the PACT fellowships, for the Empower fellowships, and hoping that we can grow these collaborations across Africa and work on this microbiome project together. So yeah, if anyone, as the others have said, is interested in collaborating, also very happy to, if you want to contact me. Thank you.
We have around 6 minutes and may be able to discuss some topics and very quick questions and answers. Let me start with Dr. Ndolo. Could you just tell us what are the challenges that are currently affecting and preventing the widespread registration of biopesticides and the use of these products, please?
Thank you very much, Luiz. So there are a couple of challenges facing biopesticide research, development, and adoption by farmers. One is regulatory challenges. So many countries still rely on a regime that was developed for regulating chemical pesticides. And those do not always work very well for biopesticides, because for most biopesticides, you're regulating biology, living things, while for synthetic chemical pesticides, you're looking at the chemistry. And in addition to that, also you find that if you use regulations that are developed for chemical pesticides, most biopesticide companies are still quite small companies. So it's very expensive for them to be able to generate some of the information that is normally expected to be generated for, say, companies registering chemical pesticides. So to cut a long story short, one is regulations. Two, we also have issues of technical issues. So for instance, in my talk, I said biopesticides do not have the same efficacy, say, as chemical pesticides. So it's issues of or even viability. So you find that many farmers may be resistant to use them because they do not give, the immediate kill like they would see when they use synthetic chemical pesticides. So technical issues are another issue. And then lastly, it's also information. So many farmers still would not know or do not have information on how to integrate biopesticides together with conventional pesticides. You remember in my talk, I said that using biopesticides alone would not be a solution. So they have to be integrated with synthetic chemical pesticides. But there has to be information that would enable users to know how to integrate them to achieve optimal results. So I think for me, those are the three main issues. There may be a few others, but those are the three main issues that may be a problem at the moment.
Thank you very much, Dennis. Dr. Venturi, please, can you just tell us briefly how probiotics, biofertilizers, can help the growth of sorghum, for example, which was one of the topics of your talk?
Yeah, thank you, Luis. This, what we believe, also following what Lara said, what we believe is that, and also what Dennis is saying, that the use of probiotics, so doing microbiome engineering, understanding the microbiome and using this information, for example, to devise ways, a very important application is to provide to the farmers seeds which are coated with one or more microbes. We are studying bacteria with bacteria that are which form a symbiotic interaction with sorghum. This is what we are aiming to provide the farmers with seeds which are coated with 2 or 3 bacteria which sorghum likes, and it can help sorghum to acquire its nutrients. For example, sorghum needs a lot of nitrogen. So following from Dennis, to be realistic, we're doing intensive agriculture. We need to produce a lot of food. So it will probably be integrated, with fertilization, for example. So by using seeds which are coated with some beneficial bacteria, we can tell the farmer, use these seeds, and reduce your, for example, nitrogen fertilization by 10, 15, 20, as much percentage as we can. Obviously, we have to do the experiments to show how effective our probiotics are and how much we can tell the farmer to reduce its chemical input. So a very important point in the future is to find, through our microbiome studies, to find the keystone, the hubs, the principal players of the microbiome of sorghum, and then to see how effective we can use them in order to reduce the use of fertilizers. And in a similar way, we can also find microbes that antagonize disease and coat the seeds with antagonist biopesticides. They can also be applied by spraying, or there are other methodologies, putting them in the soil. But at the end of the day, I think we need to be careful, and we need to find an integrated way in order to reduce our chemical inputs. Okay, thank you.
Lara, could you please, we are running out of time, but if you have a quick answer for that, how important in this scenario that Vittorio just said, how important is to consider farming system for the microbiome research?
Thanks, Luis. I think it's important. We know that farming systems affect the microbiome, but I don't think that we understand this properly in an African context. So I think that there's smallholder farmers in Africa also have different styles of farming. A smallholder in South Africa might be quite different from a smallholder in Kenya or somewhere else. And there may be solutions, it's just knowledge-driven solutions. How do we inform better farming practices that aren't technologically advanced, that can complement, the producing biofertilizers, biopesticides, also just generating knowledge how the farmer can have an impact. But I don't think we understand African farming systems well enough, and I think that we need to integrate that into the research that we do.
Thanks. And lastly, Shams, I'm just going to go back to the topic of this section, which is South-to-South collaborations. Can you just say how important is South-to-South collaboration and how they could impact the biofuels market, for example, not just the market, but the research?
Thanks, Luis. I see that, the biofuel, specifically focusing on the biofuel, is very important. See, the agriculture is, not only prime to India, but many other countries, including Africa. And I see that there's a lot of biomass that are generated. They are common between South to South countries. And these are the biomass which are non-food biomass, so that we keep alive our food security. So the non-food part of the biomass can be converted to the biofuel. And their focus including the reducing cost for production of biofuel from those non-food biomass part is going to be a very, very important component for South to South partnership.
Great, thank you very much. With this, I will end this section. And again, thanking all our speakers and the BioAfrica for inviting ICGEB to run this section. Thank you. Bye-bye.
Thank you very much.
Thank you.
Bye.
Bye.